Good evening, students. In today's class, we are going to deal with biomolecules. The continuation of protein will be dealing with lipids and will be dealing with nucleic acids. So, first, structure of proteins. We have seen in the earlier class that various structures have been seen based on the folding of the protein: the primary, the secondary, the tertiary, and the quaternary. We will be dealing with them in detail today. Okay, so first is the primary structure of proteins. We saw that in the primary structure, the linear chain of amino acid is present. Now these amino acids are linked by what type of bond? Hey, peptide bonds, right? Peptide bonds. Okay. Now the question arises here is, from where did this linear chain came from? From where it came? From DNA sequences. The DNA underwent transcription and it produced RNA. This RNA underwent translation and it produced amino acids. Okay. Now these amino acids formed a polymer by peptide bonds, and as a result, what you got is you got what? The primary structure of protein. Okay. Now this primary structure has two ends, right? One is your N terminal, the other is your C terminal. What does it mean? The N terminal suggests us that one of the side is of the amino group and the other end is of the carboxylic acid group. Okay, so this is what the primary structure is. The primary structure is not considered the overall structure of the protein. It underwent more and more number of foldings or complexity, okay, to gain its function. For a protein's function, what is responsible? Its structure is responsible, okay, its shape is responsible. We know that enzymes are functional proteins, right? We will be dealing with enzymes separately, but we know that enzymes have particular regions of actions, right? These are considered to be active sites. So active site shape is important for its function, how the enzyme is going to function. Okay, in the same way, for all of the proteins, their structure and shape is important. Primary structure is a linear structure. One end is the C terminal, the other end is the N terminal. Okay, so again, the peptide bond is nothing but a covalent bond only. It is planar, it is rigid. Okay, the order of amino acid, in which order the amino acid is in the primary structure, is determined by the DNA sequences, nucleotide sequences, ATGC, all those things. Okay, so sequence of the gene encoding the protein. We know that there are different types of within the mRNA molecule which undergoes translation has different types of codons, right? One is the start codon, which codes for methionine. So these are responsible for the production of the primary structure of the DNA. Now let us move on to the secondary structure of the DNA. We have seen 3 examples. We will be covering 2 examples. One is your alpha helix, another is your beta sheets, and this is your collagen helix. Okay, Collagen helix is also a form of a secondary structure only. Okay, so now first we'll be dealing with our alpha helix. So what is an alpha helix? The amino acids form a right-handed coiled structure. Right-handed coiled structure. Okay, this particular structure of protein is considered to be the alpha helix. What happens here, sir? It is a right-handed coiled structure of a polypeptide chain which are stabilized by what? Which type of bonds? Hydrogen bond. The structure is responsible due to hydrogen bonds. Where is the hydrogen bond happening, sir? Hydrogen bond is happening between the carbonyl groups oxygen and the amide groups hydrogen. Okay, carbonyl group means what I mean, the COO group, right? C terminal la irukra COO group. Okay, in that one oxygen is attracted to the hydrogen of the amide group NH2, not the adjacent amide group. The amide group four residues ahead. Residue means amino acid. So oxygen of a carbonyl group will attract to the hydrogen of the amide group which is 4 amino acid before. That is what it means. Okay, so this is what is your alpha helix structure. Very important point here to notice, each turn of the alpha helix contains approximately 3.6 amino acid. This is a turn, right? This one is a turn. This one is a turn. This one over here, how many amino acids are there? 3.6 amino acids are present. Okay, the structure is compact with R groups projecting outward. The R groups, that is the side chain. We remember in alpha amino acid there is a chiral carbon at the center. You have a H group, you have a COOH group, you have a NH2 group, and you have a R group side chain, right? This side chain is projected outward so that it minimizes the clashes which might occur due to the coiling. Okay, so this is one of the important point. And sir, what are the examples to note here, sir? Keratin, the protein which is present in our hairs. Okay, it is a fibrous protein. What? Fibrous in nature. In the same way, myosin, which is the muscle fiber, again a fibrous protein. Tropomyosin, a cytoskeletal protein, giving strength and elasticity. Got it? So this was your alpha helix. Again to recap, what did we see? It is a right-handed coiled structure which is done by the hydrogen bonding of oxygen of the carbonyl group to the hydrogen of the amide group, which is 4 residues ahead. Okay, each turn is consisting how many amino acids? 3.6 amino acids. What are the examples? Keratin, myosin, tropomyosin. Got it? So this is what your alpha helix structure is. Now let us wait until you take the notes. Okay. All the examples which are mentioned here are fibrous proteins. So what analogy we can take from here is alpha helix is responsible for fibrous proteins. Okay. Okay, so I hope you have completed the notes. Now we'll be moving on to the tertiary structure of the protein. Now, when we tell tertiary structure, what shape do we mean? For secondary, I just told that it was your fibrous thing. Tertiary is nothing but your globular form. Okay, globular form. Okay, so as you can see, this forms a globular structure. Okay, set. So tertiary structure, which is considered to be the globular form, okay, refers to the overall 3D folding of a single polypeptide chain stabilized by multiple interaction among the side chains. The side chains have multiple interaction, not just hydrogen bonding. Many types of bonds are seen here. This helps in the production of a globular structure. Okay. Sir, what is those stabilizing interactions, sir? One is your hydrogen bond again, ionic interactions, hydrophobic interactions, van der Waals force, covalent disulfide bond. The second most strongest bond within a protein structure. Any type of a protein structure, the second most strongest is your covalent disulfide. So first strongest is peptide bond. Okay, so collectively determine the tertiary structures stability. These are the types of interactions which we have. Sir, this tertiary structure or the globular form is seen in which example, sir? It is seen in enzymes. For instance, we have lysozyme, transport proteins like myoglobin, signaling proteins. Okay, all these are examples of your globular form. Okay, let us wait until you take notes. Now we have missed one thing, that is the in the secondary structure we did not discuss regarding the beta plated sheets. Before going to the next topic, let us just understand what is a beta pleated sheet. Before dealing here, let us understand the alpha helix again. Now the bond which is happening, we told that a carbonyl group's oxygen will bond with four groups ahead amino acid amide group, right? With the hydrogen. Okay, so this will be the hydrogen bond. Okay. Now this hydrogen bond is happening in the same polypeptide, right? Same polypeptide chain. But in beta pleated sheets, what happens is you have opposite strands of polypeptide. Now these opposite strands will have the hydrogen bonding occurring. Okay, so how is the structure, sir? It is a folder structure which shows a zigzag pattern. Okay, so two opposite strands. Okay, now suppose there is a carbonyl group here which is having a oxygen, and suppose here is a amide group which is having a hydrogen. Okay, between this oxygen and between this hydrogen there will be a what? H bond. Okay, so for beta pleated sheets also, what is responsible? Hydrogen bonding is only responsible. The only difference here is the hydrogen bond is happening or occurring between two opposite strands. Okay. So what is the example, sir? The example to note here is the protein called fibrinoin. What? Fibrinoin, which is seen in silk. It is considered to be your silk fiber. Got it? Hmm? So this is your beta pleated sheets. Such hydrogen bonds are seen at every level, not just at one place. Okay. So this is your beta pleated sheets. You have seen the example, you have seen the pattern, you have seen the pattern of hydrogen bonding also. Okay. Take up the notes. It's beta, not just B. Okay. Beta. Okay. So now we'll continue with the tertiary structure and its interactions. Okay. We saw that how many types of interactions is seen? Hydrogen bonding again, ionic interaction, hydrophobic interaction, van der Waals force, disulfide bridges. Okay. We'll be dealing with them separately again. Okay. So how the proteins stay folded due to the interactions which we just saw. Now we'll understand them one by one. Okay. So first is your hydrophobic interaction. Remember when we discussed regarding the amino acids, we classified amino acids into polar, non-polar, acidic, basic. In hydrophobic bonding, what happens is non-polar side chains cluster inside to avoid water. Why? Because they are hydrophobic in nature. Okay, hydrophobic, and that is the reason they are considered to be non-polar too. Okay, so non-polar side chains, non-polar side chain means the R groups. Okay, cluster within the groups and avoid water, while the polar residues remain outside. What happens as a result, sir? The hydrophobic end which is coming inside, it will cause a folding to occur. Okay, so this is what is your hydrophobic interaction. Non-polar groups come inside the cluster and the polar groups go outside the cluster. Okay, in order to avoid water. That is, I am talking regarding the non-polar groups. What is the example, sir? Myoglobin core packed within non-polar residues. Okay, so this is one of the example. Hydrogen bonding, we already know that it is formed between the carbonyl groups oxygen and the amide groups hydrogen or polar side chains. Okay, it is seen in alpha helices, beta sheets. Okay. Now ionic bonds is also seen, also known as salt bridges. It is a type of electrostatic attraction which is seen between the positively charged residues. Residues means amino acids. Okay, within the tertiary structure. Positively charged amino acids. What did we discuss? Lysine, arginine, histidine. Okay, so such positive amino acids interact with negative, right? Like glutamic acid, aspartic acid, also known as glutamate, aspartate. Okay, these are negative in nature. Okay, so negatively charged. Positive negative interaction, very easy. Okay, so the strength varies with pH and the salt concentration, because again the pH controls how many ions, positive or negative, will be present within. Okay, the next is a weak form of a attraction which is known as the van der Waals force. Now this van der Waals force is occurring only due to the reason because the atoms are clubbed together, very closely packed. So closely packed atoms have a short range of interaction between them. Though individually they are weak, but they collectively add stability to the protein's core. Okay, so this is one more example. Now, pi and cation pi interactions. We discussed aromatic amino acids like, right? Phenylalanine, tyrosine, tryptophan. Such aromatic rings stack above one another. Okay, this is known as your pi interaction. What happens? Positively charged amino acids like lysine and arginine, they interact with such aromatic rings. Okay, so altogether it is known as a cation pi interaction. Okay, now at the end we have the most important, the second strongest bond which is seen in the amino acid, no, that is in the tertiary structure of proteins. Okay, but this disulfide bridge will occur only if there is a sulfur containing amino acid present, specifically cysteine. That too, not only one cysteine, at least two cysteine or more is also okay. At least two cysteines. Okay, so the cysteines will form the disulfide bridges, acting like a molecular staples. Example, we have insulin and keratin. Okay, both insulin and keratin are containing cysteine within them, and they form your disulfide bridges. Sir, how disulfide bridges is formed, sir? Suppose there are two cysteine. This is one cysteine, this is one cysteine amino acid. They have a SH group. Here also there is a SH group. Okay, what happens is the H, 2H, and by the removal of water, that is by hydrolysis, this becomes from SH to, okay, so this is what is your disulfide bridges. What is important point to note here is it occurs only in those amino acids which are containing sulfur. Okay, example, cysteine. Got it? So, shall we move to the next? Yes. So, quaternary structure of proteins. What is quaternary structure, sir? Multi-subunit assembly. With examples we'll be dealing. So, not just one polypeptide, but different two or more polypeptides come together and form a overall structure. This is what is your quaternary structure. So, refers to the arrangement of two or more polypeptide chains into a overall functional protein complex. Okay, again the stabilizing interactions, that is the attractions which are taking place, are the same which we just saw now. Okay, hydrogen bonds, ionic bonds, hydrophobic interaction, van der Waals, disulfide bridges. Now let us distinguish them into two groups. Suppose the polypeptide chains are having similar subunits. Okay, so proteins composed of identical polypeptide chains. Example, lactic dehydrogenase. Okay, now dissimilar subunits which are consisting of non-identical polypeptide chain, we have hemoglobin, okay, which has two alpha and two beta chains. Okay, and insulin A and B chains which are joined together by disulfide bond. Okay, sir, you just now mentioned insulin in the tertiary structure. That is, the tertiary structure further complexes into quaternary structure. But as a whole, insulin is considered to be a quaternary structure only. Okay, hemoglobin, insulin, lactic dehydrogenase. In lactic dehydrogenase, there are similar subunits, not dissimilar. And in hemoglobin and insulin, we have dissimilar subunits. Quaternary structure enhances stability, allows cooperativity, oxygen binding in hemoglobin, and creates functional complexity in multi-domain proteins. Okay, so this is what your quaternary structure of proteins is. You can have a note until we move on to the next topic. Okay, so now let us move on to the next topic. So as we just discussed, this is your primary, secondary, tertiary and quaternary structure. Now let us deal with the classification of proteins. Last time we discussed it, but we did not mention the examples. Okay, so classification of proteins. Proteins are classified into simple, compound and derived proteins. Sir, what is simple protein? Composed of only amino acids. Only amino acids. Upon hydrolysis produce purely amino acids. Example, albumin, globulin, glutenin, prolamine. Seringla? Seri. Compound proteins, protein plus a non-protein prosthetic group. Hydrolysis yield both amino acid as well as the other compounds. Example, we have nucleoproteins, glycoproteins, lipoproteins. Okay. Derived proteins, products of protein hydrolysis, denaturation. Example, peptones, proteoses, peptides. Okay, so this is the classification of the amino acid. Now simple proteins, albumin is a water soluble which gets coagulated by heat, found in egg white, blood serum as well as in milk. Okay, albumin, the simple protein. And globulin is insoluble in water, soluble in dilute salt solution, is present in blood plasma as well as in seeds of plants. Okay, glutenin is insoluble again in water and in the salt solution, but is soluble in dilute acids or alkalis found in wheat, glutenin. Okay, sorry. Prolamines, they are soluble in 70 to 80% alcohol. Storage proteins in cereals like zein, important example. For example, zein is seen in corn, gliadin is seen in wheat. Okay, so prolamines are nothing but storage proteins which are seen in different crops. In corn, zein, in wheat, gliadin. Okay, so this is your simple protein and its examples. Now we'll move on to compound proteins, structure and function. Nucleoprotein, nucleic acid is present with the protein. We have chromosomes, ribosomes, also viruses are included here. Okay, chromoproteins, protein plus a porphyrin pigment. Okay, it could be hemoglobin which has the red pigment, cytochrome, chlorophyll. Okay, so whenever you get this chromo means color, right? So it talks about pigmentation. Easily you can remember hemoglobin, cytochrome, chlorophyll. Glycoproteins, The name glyco suggests sugar. So protein plus carbohydrate, okay, but the amount is less than 4%. Examples is mucin, blood antigens, follicular stimulating hormone, luteinizing hormone. Okay, lipoproteins, protein plus lipid. Example plasma membrane, lipovitamin. Okay, so these are examples of compound proteins. Sorry. More and more compound proteins, phosphoproteins. Prosthetic group is phosphoric acid, H3PO4. Carcinogen, milk. Pepsin, digestive enzyme, which is present in the gastric juice. Phosphatin, egg. Metalloproteins, metallo the name suggests the prosthetic group is a metal ion, enzyme with cofactors. Ferritin, which is responsible for iron storage. Hemocyanin, copper. Okay, and in the same way, mucoprotein, the prosthetic group is a carbohydrate. Okay, greater than, for example, synovial fluids. Osteomucoprotein in bones. Osteo itself means bones. Chondromucoprotein means cartilage. Okay, so these are your compound proteins. Special proteins important to note. I'm telling you, please note it down. Keratin is a scleroprotein, has fibrous, it is fibrous, tough, resistant to digestion, abundant in cysteine. So the amino acid cysteine is abundant in keratin. It provides hardness found in hair, also in nails, horns of, for example, cows, buffaloes, and also in feathers of birds. Okay. Then we have collagen. It is the most abundant protein in the animal world. Most abundant animal protein is collagen, found in the connective tissues, bones, tendons, skin. Okay. Then we have the most abundant protein in the entire biosphere. Key enzyme in photosynthesis. Okay, what it is? Ribulose biphosphate carboxylase oxygenase. Okay, CO2 fixation in which cycle? C3 or C4? C3 cycle, also known as Calvin cycle. Okay, very important points. You have to take the note. Okay. Then we have our keep. So the one more, some extra points to consider for NEET. Okay, eukaryotic organisms, the protein consists of L conformation of amino acid. Again, what is L conformation? It is not levorotatory. It just suggests us that the one of the functional group, that is the alcohol group, is present on the left side. Okay, and while bacteria and antibodies, bacterial proteins and antibodies have D conformation. Okay, opposite to that structure. Okay, now protein properties depend on amino acid sequences and its configuration, that is its arrangement, whether it is primary, secondary, quaternary, okay, tertiary, quaternary. Even small changes can dramatically alter its function. Okay, now peptidyl transferase, the enzyme catalyzes peptide bond formation. We discussed this last class. Okay, so peptide bond is formed due to this enzyme. Okay, it is a enzyme. This enzyme is crucial for protein synthesis, which is acting on the ribosomes. Dry weight composition differs between organism. Animal cells 60 to 70% is proteins, whereas plant cells 80% is considered carbohydrate. Now we are not talking about the whole concentration of dry weight, means without water. Okay, any cell, be it animal cell, plant cell, Kandipa consists of more amount of water only. If the water is removed, then in animal cells 60 to 70% protein, and in plant cells 80% carbohydrate. Okay, so 20 amino acids, out of them 10 are essential, 10 are non-essential. 4 structural levels: primary, secondary, tertiary. Main types are simple compound derived proteins. Okay, so by this we end our proteins. Now we will be moving on to lipids. Okay, so let us start. What are lipids? So lipids can be considered to be fats, oils, right? Lipids are fats that are derivatives and their derivatives, fats and their derivatives. The term lipid was coined by Bloor. Okay, it is also an organic compound. It contains again carbon, hydrogen, and oxygen like carbohydrate. But the thing to note, the difference between lipid and carbohydrate is oxygen content in lipid is significantly lower, and we already discussed that lipids are considered to be macromolecules only due to the reason that they are part of the acid insoluble pool, right? But their molecular weight is not above 800 Dalton. It is less than 800 Daltons. Okay, even though it is considered as a whole, not alone like a fatty acid. As a whole, a lipid is considered to be a macro form of a molecule, but its molecular weight is less than 800 Daltons. Now, it is soluble in organic solvents only. If you see, it is insoluble in water. It is insoluble in water. Organic solvents like alcohol, ether, okay, and it is only 3.5% of the cell content. Okay, so what it is a fuel reserve of the body. More than carbohydrates, the energy stored in the body is in the form of lipids or fats. Okay, so it is the constituent of the membrane. We already discussed. Phosphobilipid layer. Okay, they are source of fat soluble vitamins, metabolic regulator, insulating materials, also one more, shock absorbers. Okay, also are considered to be shock absorber. They are found as small globules in protoplasm, do not form polymers, provide double the energy of carbohydrates upon oxidation. Okay, so why it is important? It is an energy powerhouse, as we saw just now. Provides double the energy than carbohydrates. Storage efficiency. Lipids, though, since they are hydrophobic, they condense within the cell, not soil, occupying very less space within the cell. Okay, so during storage as compared to carbohydrates, animals store maximum food as lipids. Metabolic water source. Lipid oxidation produces maximum amount of metabolic water compared to carbohydrates or proteins. Protection and insulation, as we saw, it is a shock absorber and a food reservoir, and also provides thermal insulation. We know that fat people or a fat organism can survive even in extreme cold environments, right? So it provides survival in cold environment. Okay, understood? So it is an important point to note. Now the lipids are categorized into three: simple, complex, also known as conjugated, and derived. Okay, so fats and oils, waxes, phospholipids, glycolipids, lipoproteins, steroids, terpenes. Okay, now this simple lipid is also known as a neutral fat. It is a long chain of fatty acid, a carboxylic acid, okay, is combined with a alcohol. Okay, so fats are nothing but esters of alcohols and fatty acids. Okay. Now this alcohol is usually a glycerol, and if it is a glycerol, then such a fat is known as a true fat. Okay. When the alcohol group is a glycerol group. Okay. It includes fats, oils, and waxes. Conjugated lipids, that is, it is combined with a non-lipid group. Lipid combined with a non-lipid group. Phospholipid, phosphate group, glycolipid, carbohydrate, proteins, protein group. Okay. And derived lipids, complex structures derived from simple or conjugated lipids. So they are derived, they are derivatives of either simple or conjugated lipids. One more point to note here is they do not have a specific thing which I will be telling you by the session. Okay. Sorry. So includes steroids and chromolipids. Chromolipids is also known as terpenes. Okay. So terpenes. Simple lipids. They are, as I said you earlier, esters of fatty acids with alcohol. They are of two types: neutral or true fats. Then the alcohol group is a glycerol. If it is other than glycerol, then it is considered to be a wax. Okay. So simple lipids. Just a minute. This neutral fats are made up of carbon, hydrogen, oxygen, but oxygen is less, as we already see. Glycerol and fatty acids are the two main components. Okay. So as you can see here in this diagram, this is the structure of glycerol. CH2OH, CHOH, CH2OH. Carbons are connected. So this is your glycerol. Now, if in this alcohol group a carboxylic acid is joined by the formation of ester bond, if only one carboxylic acid is present, I will write it as one carboxylic acid, only one is present, then it is considered to be a monoglyceride. So there is a head and there is just one tail. This is your monoglyceride. Now suppose one more alcohol is attached to one more carboxylic acid. This is one more carboxylic acid. Then it is considered to be a diglyceride. Okay. So first alcohol, only one mono, two alcohols having equally one carboxylic acid by the formation of what bond? Ester bond. Okay, ester bond. It is considered a diglyceride. If again this alcohol group is also having a carboxylic acid, then you can easily guess it is a triglyceride. Okay, triglyceride. Okay, so for a diglyceride, the structure would be, I've seen it in the lipid membrane, right? Lipid bilayer. Two tails are there. We'll be dealing it separately. In triglycerides, there are, okay, this is not a jellyfish. Okay, it is a triglyceride. Okay, now fatty acid could be a saturated or a unsaturated fatty acid. Now let us understand them more in detail. Triglyceride we have understood, okay, how it is formed. So fatty acid, saturated versus unsaturated fatty acid, very important because the examples are important. Say, saturated, the hydrocarbon chain is saturated with hydrogen because all bonds are single bonded structure. Okay, no double bond is present. We have seen in NCERT 10th itself, right? Saturated Carbon compounds are those which have only single bonds. Same concept. Okay, single bond means saturated fatty acid. Two important examples. Palmitic acid. How many carbon it is containing? Here it is 14 plus 1 plus 16 carbon molecule. What? Palmitic acid, saturated. Stearic acid, 16 plus 2, 18 carbon. 18 carbon is present in stearic acid. Now this saturated fatty acids are solid at room temperature. Tell me an example we daily use it and use in our homes, but is not recommended by the doctor. What it is? Ghee, butter. Okay, these are solids at room temperature, right? These are saturated fatty acids. Less reactive, tend to accumulate, causing obesity. Okay, unsaturated fatty acid, single bonded here, at least one double bond contain one or more double bonds. Most important examples: oleic acid, linoleic acid, linolenic acid. Okay, oleic, linoleic, linolenic. Liquid at room temperature, more reactive, less reactive, more reactive. These are actually metabolized for energy. Doctor advises to always consume what type of oils? Unsaturated. Moreover, saturated fatty acids are considered to be fats, but unsaturated is not at all fats, it is oils, due to the reason because it is a solid and it is a liquid. Okay, called essential fatty acids because it cannot be synthesized within the body. Got it? So this was the difference between the saturated and the unsaturated part. Now if the unsaturated part is a polymer, then it is considered to be a polyunsaturated fatty acid, right? Example: arachidonic acid. Okay, this linoleic and linolenic acid. Medical recommendation by doctors is polyunsaturated lipids is advisable for patients who is having high blood cholesterol and cardiovascular diseases. Which is advised? Polyunsaturated. If he consumes saturated, that's all khatam. Seri. Because it lowers blood cholesterol levels. It increases only HDLs. What is HDL, sir? High density lipoproteins. High density lipoproteins. We will be discussing it separately. LDL versus, now this polyunsaturated fatty acid, that is oil, increases what? HDL. This is good or bad? It is good. Okay. If LDL is increased, that is LDL. LDL is bad. Low density lipoproteins. It is bad. Okay. So this was the clinical significance. Now waxes. These are again what? Simple lipids. So long chain saturated fatty acid, does it contains glycerol? No, it contains an alcohol any other than glycerol. Glycerol is not in waxes, any other form of a alcohol. Examples we have beeswax. It is a wax. It is secretion of abdominal glands of worker honeybees. Abdominal glands of worker honeybees. That is what we consume. Okay. Lanolin or wool fat, secretion of cutaneous glands and obtained from wool of sheep. Sebum, one of our part, secretion of the sebaceous glands of the skin. This sebaceous gland is mostly found on our faces. That is why it is most of the time oily in nature. Okay. Cerumen, soft and brownish waxy secretion of the glands in the external auditory canal. Okay, also called as earwax. Okay, we always get it. Then we have plant wax, coating formed on the plant organs to prevent transpiration. We have seen in ninth standard cuticle, right? Hmm? Cuticle, wax, all those are considered to be plant wax. Paraffin wax is not a natural thing. It is an artificial thing which is obtained from petroleum. Okay, so these were the types of waxes, and this beeswax is composed of hexacosyl palmitate. Okay, then you have carnauba wax, which is myricyl cerotate, which covers leaves, stems, and fruits. Maximum in xerophytic plants to prevent water loss. What it is? Carnauba wax is composed of myricyl cerotate. Sorry, spermate found in whale and dolphins skulls, used historically in cosmetics and candles. Okay, the part of a dolphin or a whale. Okay, cerumen ear wax, we have already seen it. External auditory meatus, protects and lubricates the ear canal. Okay, now we'll be dealing with the conjugated lipids. It has many types. We have seen phospholipids, lipoproteins, we saw, right? First we are going to deal with phospholipids. Phospholipids we have seen earlier in cell biology, cell the unit of life, as which topic? Plasma membrane. Which model? Fluid mosaic model. Fluid mosaic model was introduced by Singer and very good, Singer and Nicholson. Okay, sorry. So phospholipids, what it consists of is, it consists of two fatty acids, right? How many fatty acids? 1, 2. Okay, the tail, how many tails are there? 2. 2 fatty acid molecule. The tail is the fatty acid. We just saw it, right? Monoglyceride, diglyceride, triglyceride. It is a diglyceride. Okay, it has a glycerol. Phospholipid plasma membrane has a glycerol. To that glycerol, first 2 carbon is having 2 fatty acid. Third carbon, that is the last CH2OH, is having 2 things. Negatively charged, what? Negatively charged phosphoric acid. Okay, again from this phosphoric acid, water molecule is released when it forms a bond with the glycerol alcohol group. So H2O will be released. What will be remaining is PO. Okay, so it is a type of a, then it is also attached to a nitrogenous compound which is known as the choline group. What? Choline group. This choline group is positively charged. The phosphoric is negatively charged. As a whole, positive and negative, both at the third carbon of the glycerol, makes it what? Polar in nature, right? Water molecule is considered polar because it has both positive and negative ends. Same here. Now this is responsible for the polarity. Polarity at which level? At the head level. So this is your polar head and this is your non-polar tail. Now you understood? Okay, so choline, phosphoric acid, 2 fatty acid molecules, glycerol is the backbone. Okay, they are the most abundant lipids in the protoplasm. Which? Phospholipid, because all together in the membrane of all organelles, membrane of the whole cell itself. Okay, they are considered amphipathic. Amphipathic means again what? Polar and non-polar nature. Hydrophilic, polar end. Attracts what? Water. Water is also polar. Head is also polar. Polar and polar got attracted. Attracts water. Okay. Hydrophobic non-polar end, which is consisting of fatty acids. So since both ends are there, that is why this is known as amphipathic in nature. When we discussed regarding amino acid, we used one more term, that is amphoteric nature. So you have to not confuse yourself with amphipathic and amphoteric. Amphoteric means it has what? At one end there is a NH3 plus, other end CO minus. This makes it what? A Zwitter ion, right? Huh? Zwitter ion is what in nature? Both acidic and basic at the same time, right? So it can act, that is, it can act either as an acid, it can act as a base. Okay, so that is why it is considered to be amphoteric. Amphoteric means a substance which can act both as an acid as well as a base. Amphipathic means it has a polar and a non-polar end. Okay, got it? So now we'll be discussing. This is the diagram of what? That is the phospholipid. This is your polar group. That is the choline group. Okay, which is also having your phosphate group. Altogether, it is polar in nature. Then you have your glycerol. This glycerol is attached to two fatty acids. This is the hydrophilic head, hydrophobic tail. Okay, this is what is written here. Okay. Now, types of phospholipids. Phospholipids what are types? Types: lecithin, phosphatidyl choline, which we just saw. That structure is lecithin. Now, lecithin is found in egg yolk, oil seeds, and blood. Within blood, it is acting as a carrier molecule. Questions were asked previous year regarding lecithin as a carrier molecule for transporting other lipids within blood. Okay. Cephalin, again a nitrogenous compound, and the nitrogenous compound is ethanol amine. There the nitrogenous compound was choline, here it is ethanol amine. It is similar to lecithin. It is present in the nervous tissue, egg yolk, and blood platelets. Sphingolipids, sphingomyelins, again similar to lecithin, but glycerol, which is present in the lecithin phospholipid, is replaced with sphingosine, that is amino alcohol present in the myelin sheaths of nerves. We have seen axon. Axon is having myelin sheath, right? Schwann shells. Okay. So other importance are phosphatidyl serine, phosphatidyl inositol, plasmogens. Okay, these are other important types of phospholipids. Okay, glycolipids. The name glyco suggests it contains sugar. Composition, again two fatty acid molecules, but instead of glycerol we have sphingosine, amino alcohol, and a sugar, a galactose sugar, a monosaccharide, right? It could be also a glucose. Okay. Types, very important. Question may be asked. Cerebrocytes present in the white matter of human brain. Gangliocytes, ganglio, nerve, ganglia, spleen. It contains N-acetyl neuramic acid and also glucose. See, here the sugar part is glucose. Glycolipids on cell surface help in cell recognition and signaling. Okay, so in humans, egg surface, human egg, female, is having a glycolipid which is fertilizer, no, glycoprotein. Okay, with while sperm surface has anti-fertilizer. Just an important concept. Okay, you just noted down. It's a simple protein which both action are responsible for fertilization. Okay, sorry. So this is the cerebrocyte glycolipid which we saw is present in the white matter of our brain. Okay. Second. Lipoprotein, again a type of conjugated lipid. Okay. Phospholipid over, glycolipid over lipoprotein. They contain lipids and proteins in their molecules. Examples: chylomicrons. Chylomicrons are again carrier molecules which transport lipids within our blood. Okay. We have VLDL, LDL, HDL. I talked about it, right? LDL is low dense lipoprotein. Okay. HDL is high dense lipoprotein. I have to tell you an important point here. So lipid is hydrophobic in nature, but it is an important molecule. It has to be transported within our body. Now blood is the transporting, circulating part within our body. But blood is mostly composed of what? 70% water. Lipid being hydrophobic, will he stay in this water part? No. So there is a need of a separate thing which will carry such lipids to different parts. That is what is your lipoprotein. Okay. Now this lipoprotein is of these types. Okay. That is chylomicrons, VLDL, LDL, HDL, free fatty acid, albumin complex. What is LDL, sir? It is bad for our health because its most content is lipid. Most content is lipid and the protein content is less here. Okay. So it gets accumulated, causes atherosclerosis. Atherosclerosis. What it is? It causes cardiovascular diseases. It blocks our arteries, blood vessels. Okay. Now HDL is high dense. Now this, since it is totally lipid, lipids are considered less in Dalton's, right, molecular weight. That is why low dense. But high dense lipoprotein is consisting mostly proteins. Okay. Protein content is high here as compared to the lipid content. So the density is increased. Even if lipid is going to accumulate, it will accumulate in less quantity within the arteries. It will never block it. Okay. So that is why physicians advise us to take HDL in the form of unsaturated, polyunsaturated fatty acids. Okay. So this was your lipoprotein we have discussed. Now derived lipids. Okay. Derived lipids are got from either simple or a conjugated lipid. Again, they are insoluble as a lipid. All steroids have a tetracyclic structure called cyclopentane prehydrophenanthine nucleus. What it is, sir? It is a 4 interconnected rings. 1, 2, 3, 4. Same is here. Same is here. Same is here. Vitamin D is a steroid. Cholesterol is a steroid. Cortisone is a steroid. Testosterone is also a steroid. Now the steroid is classified into two groups again based on the functional group it is having. What? If it is a alcoholic steroid, it is considered sterol. If it is a ketonic steroid, it is a sterol. Easy? Now one more, they are derivatives obtained by the hydrolysis of simple. They are obtained by the hydrolysis. Okay. What was the important point which I said you, which is missing in steroids as compared to other lipid, is that it never has fatty acids. Does not has fatty acids. It has only alcoholic groups or ketonic groups. No fatty acid at all. Okay. But are included in lipids as they have fat-like properties. Only due to the properties it has. Okay. So the most common steroid is sterol, cholesterol, ergosterol. Okay. We'll be dealing with them. So cholesterol, most abundant in adrenal gland. So what is adrenal gland? Located superior to the kidneys. Okay, each kidney. Brain also it is present, nervous tissue also on the skin. It is considered to be the most decorated micromolecule. What molecule? Not macro. Micro, because we are not discussing whole of lipid, just steroids. Okay, micromolecule in biology. Parent steroid for other biological important steroids. Okay, now we have our 7 dihydrocholesterol present in skin as provitamin. When exposed to UV radiation, it becomes vitamin D, which is also known as, question will be asked here too. Okay, now ergosterol is present in oil seeds, fungi like or ergot. What is fungi like ergot? Ergot is a fungal disease. So when this fungal disease is caused, it produces ergosterol. Okay, and also by yeast. Yeast is again a fungus. It is a precursor of ergocalciferol, another form of a vitamin D. Whenever you get this name, calciferol, you have to understand it is vitamin D. Okay, bile acids, which is produced by the bile juice, is produced by the liver, right? Now it contains different steroid acids like cholic acid, lithocholic acid, that helps in fat emulsification during digestion. What it? Cholic and lithocholic acid. So we have discussed some important points over here. Okay. Sterols. Again, it is having what? Ketonic groups. Okay. Ketonic steroids. Just to revise, it consists of sex hormones, adrenocorticoids, ecdysone. Okay, sex hormones, male sex hormone, testosterone, female progesterone, as well as estrogen. Okay, they regulate the reproductive functions. Adrenocorticoids, the name adrenal means again adrenal gland on the kidneys. Which part adrenal glands? Internal part or outer part? Outer part, which is your adrenal cortex. Adrenal cortex is the outer region of the adrenal gland. Regulate metabolism, stress response, and electrolyte balance. Ecdysone present in insects, secreted by prothoracic glands, controls molting and metamorphosis. What is molting, sir? Molting is the process by which the insect sheds its exoskeleton. It sheds exoskeleton. Okay. And again, metamorphosis is the process by which a earlier form of a insect, that is a less mature form, turns into adult. That is metamorphosis of a butterfly, right? I'll write butterfly for your better understanding. Okay, sorry. Diosgenin extracted from yam. In Tamil it is known as Maraveli Kelangu. Yam. Okay. Dioscorea. Okay. Is used in the manufacture of anti-fertility or contraceptive pills. Okay. Diosgenin. Dios, yam. Okay. It is a vegetable which is commonly used in India. Okay. Now we'll be dealing with chromolipids, also known as terpenes. Okay, complex lipids, which is made up of repeated isoprene units. What is isoprene? C5H8. Chromo, the name suggests, it gives color. Okay, crucial role in various biological processes. Carotenoids, vitamin A, E, K. Okay, essential fat soluble vitamins. What? A, E, K are fat soluble vitamins. Questions can be here. A, E, K. Okay, sorry. Natural rubber, polyterpene from rubber trees. Okay. Now you have vitamin A as lycopene, a pigment which is present in the tomatoes. Gibberellin is also a what? A terpene only, okay, which is a plant hormone. Okay, it is also a term. What we have discussed: lycopene, gibberellin, vitamin A, E, K, fat soluble vitamins, as well as carotenoids. Okay, so these are considered to be chromolipids. So, a summary of lipids: it is energy storage, most efficient energy storage, stores double the energy than carbohydrates, membrane structure, phospholipid, choline nitrogen. What that carrier molecule is called? Lecithin. Okay, sorry. Insulation, even at colder environments we can survive if we have more fat content in our body. Okay, it is also a part of the myelin sheath. Vitamin carrier. Okay, fat soluble vitamins A, D also. Okay, vitamin D also, and signaling protection. Got it? Now, by this, some important facts. Lipid provide, this we have seen. Linolenic, arachidonic cannot be synthesized in body, must be obtained from diet. So these are known as essential fatty acids. Okay. Now, amphipathic nature we have seen. Cholesterol importance, it is the precursor of vitamin D. We saw, right? Bile acid, steroid hormones. Okay. Now, clinical correlations. Atherosclerosis, already I have mentioned it. So, excess saturated fat. What type of fat? Saturated fat. And cholesterol lead to plaque formation in the arteries. What is plaque? It will block the arteries, okay, causing cardiovascular diseases like stroke, heart attack, all those things, okay. So, respiratory distress syndrome. Premature infant lack a compound called dipalmitoyl phosphatidylcholine. This is a surfactant. It causes breathing difficulties. Only due to the reason this compound is missing or is in lack in premature infants, what is caused is, it causes breathing difficulties is caused in infants. Okay. Deficiency of bile salts or pancreatic lipase leads to steatorrhea, fatty stools, okay, and vitamin deficiencies. Got it? Lipase is a enzyme, okay, which uh, breaks down lipids to fatty acids. Okay, got it? So we have discussed the most of lipid metabolism. Again, we discussed lipase enzyme breaks down triglycerides, absorption. This fatty acids which have been broken down. All this breaking down is happening in small intestine. Lipid is broken down into triglyceride, and it is further broken down into fatty acids and glycerol. Now, fatty acids and glycerol are absorbed by intestinal cells called villi. Remember? Now, reformed into triglyceride and packaged chylomicrons. We saw, right? Lipoproteins which transport lipids within blood. Storage and used, stored in the adipose tissue, okay, beneath our skin, mobilized during fasting through beta oxidation, which is done in mitochondria. Okay, got it? So this is, so we have did all these things which you can see later in the video. Now, the molecule of life, nucleic acids. Nucleic acids we have two, right? One is the DNA and the other is the RNA. Deoxyribonucleic acid, ribonucleic acid. Okay. So, nucleic acid first discovered by Friedrich Mischer. in the nucleus of pus cells. Pus cells, okay. What the name did he give? He gave the name not nucleus or DNA, he gave the name nuclein. This was the name given by whom? Misher, okay. And the nucleic acid, the name was given by Altman, okay. So, what are the components of nucleic acids? Nucleic acid is made up of three components. Three components. One is your sugar, always a pentose sugar. Okay, two types of pentose sugar. One is your ribose, or it could be or deoxyribose sugar. We already saw the difference when we discussed regarding carbohydrates. Difference between ribose and deoxy. Second carbon in deoxyribose sugar is lacking oxygen. Here it is present. Okay. Then sugar is a component. The second component is a phosphoric acid group. Okay. Or a phosphate group. Okay. Then you have your nitrogen bases. Okay. So this is the nucleic acid is having three components. Any form of a nucleic acid, whether it is a DNA or a RNA. Okay. So see, this is your deoxyribonucleic acid. This is first carbon, second carbon, third, fourth, fifth, and six carbons are five carbons only. Sorry, pentose sugar, right? The second carbon, if you see, it should have a O group in ribose part, but here it is not present. But in ribose, the second carbon is having a O. Okay, since it is lacking, it is known as deoxy. Actually, the full name is 2-deoxyribose. What? 2-deoxyribose. Okay. Now, nitrogen bases are of two types. One is your purine, the other is your pyrimidine. Pyrimidine is having only one ring, but purine is having two rings. In addition, it is having an extra ring which is known as your imidazole ring. What ring? Imidazole ring. What are the types of purines? Adenine, guanine. Types of pyrimidine: cytosine, thymine. In DNA, if it is a RNA, not thymine, it is a uracil. Okay, so you have to also know the structure. Okay, how many rings? 1, 2. Sorry. So the building blocks are actually nitrogen bases. As we discussed, it is of two types: pyrimidines and purines. Single ring structure containing 4 carbon and 2 nitrogen atoms. 4 carbon, 2 nitrogen. Types: cytosine, thymine, present in DNA. Cytosine and uracil, present in RNA. Okay, RNA. Purines, double ring structure with pyrimidine, that is pyrimidine structure is along with the imidazole ring. Okay, examples are adenine and guanine. Very important. We also saw the structure, we also saw the features of it. Now, nucleoside versus nucleotide concept. Very important concept. Nucleoside means nitrogen base when it bonds, with the first carbon atom of the pentose sugar. First carbon atom, one of the nitrogen base is going to bond. What type of bond? N-glycosidic linkage. Nitrogen at position 3, okay, forms the bond with, that is in pyrimidines. In pyrimidines, nitrogen at position 3 will form the bond. In purines, nitrogen at position 9 will form the bond. Okay. Now, nucleoside, how they are named? How they are named? The nomenclature. Okay. So, this is a nitrogen base, right? It is joined with a ribose sugar. Ribose sugar is in RNA. Adenine plus ribose becomes adenosine. Guanine plus ribose, guanosine. Cytosine plus ribose, cytidine. Next, thymidine, uridine. Huh? But if it is a deoxyribose, then what you will say? Deoxyadenosine, deoxyguanosine, deoxycytidine. Easy. Nucleotide. If the same nucleoside, nitrogen base plus sugar, is again attached, or we are considering one more group, that is the phosphate, then it is a nucleotide. A complete nucleotide forms when phosphate reacts with the fifth carbon atom of the sugar, forming what type of bond? That is why it is known as a phosphoester bond. Okay, phosphoester bond. Adenosine plus phosphate becomes adenylic acid, also known as adenosine monophosphate. How many phosphate? 1. Anything you are getting? ATP. ATP is also what? A nitrogen base. Okay, but it has three groups of phosphate. Guanosine plus phosphate, guanylic acid, GMP. Cytidine plus phosphate, cytidylic acid. Okay, so what bond is this? This bond is phosphoester bond. That is this bond. Okay, which carbon? Fifth carbon. From the fifth carbon this bond is forming, and this is your first carbon. This is what? A pyrimidine or a purine? A purine. Ninth position. Saringla? Ninth position, first carbon. What bond? N glycosidic bond. What bond? Phosphoester bond. Okay. Now one more thing to note here is, there are not just one nucleotide, many nucleotides forming a linear chain, right? So between phosphate and sugar, what bond? Phosphoester bond. But if there are many nucleotides, suppose this is nucleotide 1, nucleotide 2, there will be a phospho group. It will get connected to the next nucleotide. Now this is known as a phosphodiester. Okay, got it? So you have to remember all these. So these are different types of nucleotides. This is your, since it is a deoxyribose, then it will be called as deoxyadenylic acid or adenylate. We also that, or DAMP, damp. The D is small in the lower case. It suggests it is a deoxyribose. So easily you can now remember, right, every name. You can either write monophosphate, ilic acid, or ilate. Okay, for nucleotides. For nucleosides, INE. Okay, match the following, they will ask. Okay, mostly it is asked in match the following. Okay, set. So this is your overall structure of your DNA. How many strands are there? 2 strands. This is one strand, this is one strand. Both the strands are anti-parallel in nature. What is anti-parallel, sir? One strand is 3 prime to 5 prime, other is 5 prime to 3 prime. Okay, this is what anti-parallel is, and all these are made up of, see, this strand is containing phosphate and sugar, whereas the connection is formed between nitrogenous bases. Now these are connected by, we saw right, phosphate, that is one nucleotide and another nucleotide are connected by phosphodiester. Phosphate and sugar are connected by phosphoester. Then sugar and nitrogenous bases are N-glycosidic bonds. Nitrogen bases and nitrogen bases are also connected, right? Hydrogen bond. Okay, so hydrogen bond. Hydrogen bond. Adenine always forms, it bonds with thymine. How many bonds? 2 hydrogen bond. Guanine always forms its bonds in cytosine. 3 hydrogen bonds. Okay. In the case of a DNA. In the case of a RNA, adenine, uracil. Guanine, cytosine. Easy? Same. So the double helix structure, before that it was discovered by Misher. The term DNA, the name was given by Zaccharis. Okay, the revolutionary double helix model which we have just saw right now was given by whom? Watson and Crick. Based on, in which year? 1953. By what study? X-ray crystallographic study of whom? 2 scientists, Wilkins and Rosalind Franklin. Okay, now Wilkin, Watson and Crick got the Nobel Prize in 1962. Pavo, Franklin did not get. Okay. So key features of DNA double helix: anti-parallel strands, 2 polynucleotide chains, that is 2 strands. Okay, or both are running in opposite direction. One strand is 3 to 5, 3 prime to 5 prime, and 5 prime to 3 prime. Second, complementary base pairing. Adenine pairs with thymine with 2 hydrogen bonds, and guanine with cytosine with 3 hydrogen bonds. Sugar phosphate backbone. The backbone of DNA is sugar and phosphate. Okay. Helical configuration, double helix in specific dimensional properties. Now we will be dealing with Chargaff's rule. Chargaff's rule, very important rule. Okay. Purine to pyrimidine ratio is 1:1. In a double stranded DNA, the amount of purine nucleotides is equal to the amount of pyrimidine nucleotides. A equal to, not double bond, this is equal to. Okay, here also it is equal to. Adenine amount is equal to thymine amount. The quantity of adenine always equals to the quantity of thymine. Guanine is always equal to cytosine, the amount. Okay, now it is not necessary that A plus G amount is always equal to C plus T. Sometimes it could be different. Okay, A plus T, G by C base ratio. We'll be discussing about it. Okay, just a minute. So DNA molecular dimensions. Okay, what I'm saying, that is the pitch value per turn, how many nitrogenous bases, 1 turn the length, how much, all these things we are going to discuss. So helical parameters, distance between successive nitrogenous bases is 3.4 angstroms, or we can say 0.34 nanometer, 0.34 nanometer or 3.4 angstrom. 10 to the power minus 10, 10 to the power of minus 9. Sorry. Complete turn length 34 armstrongs. Okay, one complete turn. Okay, that is this is one turn, right? So it is about 34 armstrongs in, and per turn, that is In this one turn, how many base pairs? 10 nitrogenous base pairs. Got it? Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. That is what it means. In this one turn, how many are there? 10 base pairs are there. Are you understanding what I am saying? What we have discussed? The pitch 3.4 Armstrong. Then how many base pairs? 10. And complete turn length is 34 Armstrong. Okay. Now structural measurements. DNA's diameter, overall diameter is 20 Armstrongs. In distance between the strands, opposite strand, this distance is 11.11 Armstrong. Hydrogen bond length 2.8 to 3.0 Armstrong. It differs. Okay, very less. Molecular property, base sugar angle is, that is the angle between the base and the sugar is 51 degree. Molecular weight is 10 to the power 6 to 10 to the power of 9 daltons. That is why it is considered to be a macro or a micro? Macromolecule. Okay. Overall. Now, types of DNA, right-handed versus left-handed. Right-handed is also known as ddNA, left-handed is also known as zDNA. This was actual Watson and Crick model. Okay, clockwise turning. Clockwise turning means clockwise from left to right or right to left. And zDNA anti-clockwise. Okay, so things will be opposite here. And zigzag backbone arranged, phosphate and anti-clockwise rotation, zigzag backbone found in specific sequences only. Two types of, so this is your clockwise, this is anti-clockwise. Right-handed, left-handed. bDNA, zDNA. Okay, Z or Z, whatever. Now DNA in eukaryotes versus prokaryotes. Very easy. It is linear, it is circular. Primitive forms have circular DNA. Advanced forms have linear DNA. Okay. So eukaryotic DNA, linear chromosome associated with histone proteins. We saw, right? Nucleoprotein. Then histones occupy major groove at 30 degree angle. Salt linkages are present. Okay, with which bound DNA to histone proteins. Higher AT content, eukaryotic DNA, I told no, A and T content and G and C content. A and T content is equal, G and C content is equal, but overall AT content or overall GC content is not equal. AT content in eukaryotic is more as compared to GC content. So, okay, base ratio is greater than 1. What is base ratio again? AT content divided by GC content. So, the value is greater than 1. But in case of a prokaryotic DNA, what is more? Higher GC. When GC is more, melting point is also more. Yeah, the melting temperature due to GC bonds is higher. Okay, one more point. They are not associated with histone proteins. They are naked DNA, also known as nucleoid, or genophores. What? Genophores. What it? DNA denaturation and renaturation. Native DNA is double stranded, right? But when temperatures of 80 to 90 degrees Celsius is attained, hydrogen bonds break down and the double stranded DNA becomes single stranded. Okay. Upon cooling again, it renatures, that is, it binds again with hydrogen bonding, restoring its double stranded structure. Now, this denatured form is has hyperchromaticity. Denatured form has hyperchromaticity. It absorbs more light. This is hypochromaticity, absorbs less light. So we have seen what is denaturation, what is its property, renaturation, its property. What it? Sorry. DNA replication. So we have seen in cell division that during the S phase of the cell cycle, the content of the DNA doubles. Okay, it doubles due to the process of replication. Okay, now for replication there are different models. One is your dispersive model, one is your conservative model, and the other is your semi-conservative model. But the most accepted model is semi-conservative model. Each new DNA molecule contains one original strand and one newly synthesized strand. We'll understand it through a flowchart, don't worry. Conservative original DNA remains intact, entirely new double helix is formed. DNA breaks into fragments that are copied and randomly reassembled. Now we know that the most accepted form is semi-conservative model. How do we know? There was an experiment which was conducted by Messelson and Stahl. Okay, using CSCl, cesium chloride centrifugation method. Okay, they used a radio labeled DNA. What is a radio labeled DNA? A DNA which is labeled by a radioactive metal or a non-metal, whatever. A radioactive element. Non-metal, element. Okay. So this is what the proof for semi-conservative model is. What happens in semi-conservative model is, suppose this is your DNA strand. These both are what? Parental strands. Okay. What will happen is they will separate. Now from each strand, what will happen is new strand will be synthesized. Okay. So as a result, this will be one copy and this will be your second copy. So this is what is your semi-conservative. So semi means half is conserved. Half of the old strand is conserved and half of the new strand is produced. Okay, this is what semi-conservative model is. And this DNA replication is done by various enzymes. For example, DNA polymerase enzyme. Okay. Understood? Easy? Okay. DNA completed. Now RNA. Key difference again, it has what? Uracil which replaced thymine, and the sugar is a ribose sugar. It is more chemically reactive. If you consider RNA and DNA is stable, RNA is reactive, little bit unstable than DNA. Okay, shorter lifespans in cells. Okay, now types of RNA: ribosomal RNA, transfer RNA, messenger RNA. 80% of the total cellular RNA is ribosomal RNA. Okay, mostly. It was discovered by Kuntz, produced in the nucleolus of the nucleus. Most stable RNA form is the ribosomal RNA, structural component of ribosomes. Ribosomes are also produced, also made up of what? This only, ribosomal RNA only. Transfer RNA 10 to 15%. Okay, also called as soluble RNA, discovered by Hogland, Zemanek and Stephenson, is the smallest RNA, 4s subunit. S means sedimentation or Svedberg unit. Okay, Svedberg unit. It talks about the sedimentation coefficient. Okay, it has a clover leaf structure. What is a clover leaf structure, sir? This is a clover leaf structure. tRNA. This is your rRNA. Okay, got it? Now mRNA, 1 to 5%, the least percent of the RNA present in the cell. Huxley, Volkan and Astrachan, named by Jacob and Monod, least stable template for protein synthesis. Okay, these are the various shapes and types of the RNA present within the cell. Ribosomal RNA, so eukaryotic ribosomes are of 80S types. The subunits are 60S and 40S subunits, and prokaryotic have 70S subunits, 50S being the larger, 30S being the smaller subunit. Okay, during protein synthesis, rRNA provides attachment sites to tRNA and mRNA. Salt linkages attach to tRNA to larger subunit, mRNA to smaller subunit of the ribosome. Okay, now your transfer RNA is also known as what? The adapter molecule, single stranded hairpin loops, clover leaf structure, anticodon region for codon recognition. Okay, what codon? Codon of mRNA, genetic code, right? 3 codes we have seen, right? Okay, smallest RNA molecule we have seen, it serves as the adapter molecule during protein synthesis, also known as translation. Okay, now messenger RNA is the genetic blueprint. So DNA gets transcribed into mRNA, and this mRNA is having the genetic code. This has what? Genetic code. How many codons are there? 3 codons. Okay, each specific 3 codon is responsible for specific amino acid to be produced, right? Specific amino acid. Okay, so genetic versus non-genetic RNA. RNA could also be a genetic material. Usually in human beings, which is the genetic material? DNA. But RNA is a genetic material in viruses, reoviruses, tobacco mosaic viruses, QB, bacteriophage is also virus only. Okay, when DNA is absent, RNA functions as the genetic material, mostly in the virus family. Non-genetic RNA molecule do not serve as genetic material responsible for transcription and translation. All the 3 types of RNA. Okay, nucleotides beyond genetic information. Nucleotides in DNA we saw are responsible for carrying genetic. What are other important functions of nucleotides? ATP, energy transfer, serves as the universal energy currency of the cell. Information transfer, already we have seen, genetic material, hereditary material. Coenzyme formation, here ATP is adenosine. Adenosine means what? Nitrogen base plus sugar, right? It is having 3 triphos. Now it has become a nucleotide from nucleoside. Okay, how many phosphate? 3 phosphate. Adenosine triphosphate. You also have coenzyme functions also. For example, you have NAD and FAD. Nicotinamide, adenosine, and flavy. F stands for flavy. Okay, so these are again derived from nucleotides. NAD is responsible in what? FAD is responsible in what? Both are responsible in respiration. Okay, this in Krebs cycles and glycolysis. This in glycolysis. Okay, whatever, altogether respiration. Okay, it might be wrong, but altogether respiration. These are coenzyme function. Okay, so they act as carrier molecules. Okay, they act as carrier molecules to carry electrons. Okay, sorry. Key discoveries: Misher discovered nucleic acid in the nucleus of pus cells, named it nuclein. 1953, Madsen and Crick, double helix. 1962, Nobel Prize. RNA enzymes, Czech Altman discovered catalytic RNA. Ribozymes. What is this? You know, we didn't mention this earlier. Ribozymes are RNA molecules which act as enzymes. Important concept. What is a ribozyme? RNA which act as enzymes. Who discovered it? Czech Altman. Okay, 1981 to 83. Important thing. This was asked in previous year question paper. Okay, so that's it. We have completed protein, then we saw lipids, and we have completed nucleic acid covering both DNA and RNA. In the next class, we'll be dealing with enzymes in a separate video because it being the most important part of the biomolecule chapter. Thank you. Meet you in the next class.